Good morning to everyone. It's good to be here again. I'm not Maribel Hernandez. Maribel had a problem and I'm in his place. It's a great pleasure to present Dr. Deisy Morselli Gysi. Deisy is an associate research scientist at the Barabasi Lab at the Network Science Institute and the Northeastern University, and a research trainee at Channing Division of Network Medicine at Harvard Medical School, Brigham and Women's Hospital. She obtained her PhD in computer science from the University of Leipzig. She worked as a bioinformatician at the Heart Institute of the University of São Paulo, where she worked with co-expression networks, investigating the influence of SNPs on the networks. She also worked at the Albert Einstein Hospital as a statistician consultant. So she's Brazilian. She has interest in biomarker discovery for complex diseases related to autoimmune response in many diseases, mental disorders, and senior disorder. She also has interest in drug repurposing for those diseases, mostly using a combination of multi-omic, machine learning, and network analysis approaches. During COVID-19 pandemics, she focused on repurposing drug candidates for treating this disease. Today, Dr. Deisy will talk about drug repurposing for treating diseases, a network medicine approach. Dr. Deisy will talk to us first, and after, the participants are free to make questions to our speaker. You can write the question in the channel, we can see them, and we will read them for Deisy. Como Deisy é brasileira, vocês podem também falar em português e ela responde em inglês. Então, fiquem bem à vontade, né? A Deisy, conversei com ela agora, né? Essa palestra vai ser muito inspiradora para os jovens estudantes e jovens docentes pela trajetória que ela teve até aqui. So, Deisy, it's with you now.
Thank you so much, Maria, for the invitation, and also thank for the very nice introduction. It's a big pleasure to be here talking to this very nice crowd. So I'm going to talk today about network medicine and its application on drug repurposing. Let me share my screen. I assume you all can see this, right? So why is it important to use drug repurposing, right? And what is drug repurposing? So basically, drug repurposing means that we are using an existing drug that is approved for treating a particular disease, and we use the same drug to treat another disease. So a very nice example is sildenafil, that was first approved to treat coronary artery disease, hypertension, and angina pectoris. However, in the clinical phases, so in the clinical trials, they noticed a side effect from this drug. that the male participants, they started to have an erection when they started taking the drug. So this drug was repurposed for treating that problem, and it's now known as Viagra or vitamin V. Uh, and the same way, many other drugs had the same trajectory, right? So they are in clinical trials and they become available for other diseases, or even after the approval, they notice some side effects that can be used to treat any other disease. And why is it important for using drug repurposing in a COVID space? So we know that because of COVID, everything shut down. Otherwise, we would be in people and talking like normal today. But yeah, so because of the breakdown and everything, we needed to find drugs and in a rapid deployment, right? So that we could find an effective therapeutic intervention for treating COVID. Um, and we couldn't wait as long as a normal development where you develop a new molecule that is going to be used to treat a particular um, a particular disease, in this case COVID. So we develop a molecule that's going to be used for treating COVID, and then we do all the experimental testing, and then we go for clinical validation. You approve the molecule, you approve the drug, you approve the drug for the disease. So this long process can take from 10 to 20 years, and that's simply not feasible, not in a breakdown crisis that we are in. So a more realistic strategy is to find drugs that they are either approved or they are already in experimental setup, and rank those drugs and try to identify the ones that would have a more therapeutic effect for COVID. So for that sense, there are many different methodologies that they can be used for drug repurposing. However, network medicine has already validated a series of computational tools for identification of drugs for treating different kinds of diseases. And here, what we did is that we deployed those tools and identified a set of potential drug candidates for treating COVID. So, what else network medicine has done, right? So we started by identifying disease modules. So the disease module discovery, that's basically identifying how how genes that they are associated to disease, they relate to each other, and how can we go from that to pathway analysis, pathway identification, and which other genes that we have could also be associated with a particular disease. That also lead to drug target identification and drug repurposing. So that would be identifying possible targets for a particular drug, or simply identifying drugs that can be used for treating particular diseases. But because we know that often only one drug might not work for treating a disease, but a combination of those drugs that had led to the study of drug combinations, where you combine drugs or components from drugs, and you identify the best set of combined drugs that will work in a synergistic manner. And there is also the branch that studies personalized medicine, where you can place one particular individual in the network context and identify which is the most probable disease this patient has. And it helps with patient classification when you have diseases where it's harder to make a diagnostic just based on clinical measures. But I've been talking about networks, and what are networks, right? So networks are basically graphs where they have nodes and vertices, or links and edges. And nodes are like those big entities that we have here, and they are connected by a link. If they have some kind of relationship, and they may represent many things. So one example is in a computer network, right? So you have computers that they are connected to each other, uh, and the connection, like the link here, is they are connected, like literally connected in the same network. But we can think about this in many other ways. So because network science is almost everywhere. Uh, so imagine that we are in a party and we have multiple people in this party. So we have here 5 guys, we are all in the same party. Um, and how those people are interacting with each other can create a link here. And the interaction is what we can define as they are either dancing together, they are talking together, they are sharing a beer, they are some kind of interaction that is defined by you, right? But we can also think about this in a social media context. So we have people that they follow another one in Twitter, or they are friends in Facebook. They can be also follow another one in Instagram, or something like this. But if we, instead of be looking like in this macro social interactions, we can look into one particular individual and see how our body behaves. And that can help us understand, for example, how our brain relates to our heart, how it sends signals to our lungs, our kidney. But even if we look deeper inside that, the tissues inside our brain, they are sending different kinds of signals to different regions, right? So each part of the tissue can be related to different connections, right? And we can still look inside that and see that cells, they connect to each other in different manners. But we also have our genes that they are connected to one another, or even the proteins. So proteins, they can bind to another protein, and they can, or we can look into like pathway analysis, where one gene sends, like, has a product, and this product's going to be catalyzed by some kind of reaction, and then you have another. But we can also look into this network by a virus host interaction, right? So here we have the case of EBV, Epstein-Barr virus. So in red, we have proteins that they are from the virus, they live inside the virus, right? And once they get inside the host body, so humans, so those proteins, they're going to bind or hijack a couple of proteins, right? And those are the orange ones. However, the orange proteins, they are also connected to some other proteins. So they bind to other certain human proteins that they can be related to some diseases, right? So what happens is some of these proteins that they are targeted by the virus, they can be related to some proteins that they are associated with some diseases, and they can lead to those symptoms, right? So and following more or less this idea, we wanted to work with COVID. So I talked a little bit about protein interactions, and we saw from the previous slide that, like, the the targeted proteins, they also had some other proteins in the human body that they interact. But what I'm talking about, right? So those protein interactions means that one protein binds physically to another protein, so they connect to each other, and that's what we use. So we have a map of physical bindings from those proteins, and that can come, they often come from mining scientific literature, and those interactions, they can be based on predictions, or they can be based on experimental systematic approaches, right? And often what is used is hybrid to highest mapping, And that's we can have like a binary interaction at the end of the story saying like, oh, protein 1 binds to protein 2. And that's what we use as our map. This map looks more or less like this. So each one of the nodes that we have here, each one of the nodes here, they are the white dots. And we have a link if two proteins are interacting with each other. So this is the human interaction, right? One thing that is, like, a couple of things that they are important to know here is the interactome is incomplete, so we don't know all bindings that they exist. We know that those bindings, they can be specific to certain time, they can be specific to certain tissues, they can be specific to certain moments that the cell are passing through. So those are things that we have to keep in mind when we are working with a protein-protein interaction map. And that leads us to disease modules, right? So we have genes, when they are localized in the same topological community, they are often related to the same biological process. So if we have, like, from one side, the story, right? So we have genes that they are associated with biological process, they are in the same neighborhood, but we also know that genes that they are associated with the same disease, they are often also involved in the same biological process. So that probably means that if we have genes that they are in the same associated with the same disease, they will be living in the same neighborhood. So let's imagine, so this is the map of Manhattan. And we have specific neighborhoods there, right? So if you want to get some Oriental food, some Asian food, you probably would go to Chinatown. So if you want to go to the theater, you would probably go to Broadway. And our protein-protein interaction works more or less in the same way, right? So we have those neighborhoods, and they would be associated with some closely related things. So here we have the PPI, but we have exactly the same map as we had before, but now we have it marked for asthma genes. So in pink, highlighted in pink, we have genes that they are associated with asthma. And we can see that most of them, they are connected and they are living in the same neighborhood, right? We still have a couple of genes that they are outside of this big blob, and there are some reasons for that. So remember I mentioned at the beginning that the PPI is highly incomplete. So we estimate that now we know around 50% of the whole interactions that we might have. And we also don't know all the genes that they are associated with a particular disease, right? So we don't know all genes that they are associated with asthma in this case. And that probably leads to the separated nodes around the network that they are not connected. But if we knew the ones that they are in between, probably they could also be associated with the disease. Or if we knew more connections that we have, they would probably get tied all together. Okay, so let's go back to the COVID story. So in March 23 last year, we Gordon et al., they released the full systematic scope where all the 26 proteins from COVID, they would be binding to 332 proteins in the human body. We already knew the human interactome, and we already knew all the drugs and with their targets. And we wanted to find drug repurposed approaches that could really find something that could help us go home, like not stay home all the day. So in April 15, we finalized our predictions. And based on that, we immediately started our experimental screening, but we first validated our results in clinical trials. So we have two sets of drugs that we tested in vitro. So the first one, we have 918 drugs, and the second scope, we had 74 that they were all picked by a clinician. And we have different methods for rank aggregation and validating our results. So when we look into the disease model, the first thing we expect is that, similar to asthma or any other disease, those proteins, they would be highly connected or be close in the same neighborhood, right? So that's actually what we find. So out of the 332 proteins which SARS-CoV-2 binds in the human body, 208 form a connected component. But the first thing we have to do after doing this is trying to identify if this model is just there by chance or it's a real model. And actually, we find that it's a real model. It's greater than would be just by chance. So if we want to find a drug repurposing candidate, those drugs should be getting close to this neighborhood, right? But there is another thing that we also wanted to say is, because we know that a disease to manifest itself in a particular tissue, it has to have the majority of its protein there and have also a connected component in that part. And we actually found that there is a connected component in the lungs, where 180 proteins were present. But we also wanted to see which other tissues could be affected by COVID. And one thing that we have to keep in mind here is that we did all these predictions by March last year. So we knew very little about the disease. We knew very little about which tissues could be involved or any of the comorbidities or side effects that you could have from the COVID infection. So we found some involvement with many brain regions, and that came later to know, like, we have a lot of neurological manifestations. We have the loss of smell, loss of taste, headache, dizziness, and so on and so forth. We also found that the digestive system is also affected. So we have diarrhea, vomiting, abdominal pain, that they are all symptoms of the disease. And we also know that some cardiovascular tissues are also involved. And now we know that patients that they have any underlying cardiovascular disease, they have increased risk of death. And similarly, we also found reproductive system tissues involved, and that was really shocking for us. But we know nowadays that, for example, the testes, it acts as like a big bag where the virus is to reproduce itself. Okay, so we know that proteins that they are living in the same neighborhood, like proteins that are associated to a disease, they're going to be living in a closer neighborhood. But do proteins that they are associated with similar disease are also living in the same neighborhood? Yes. So if we have two diseases that they are very similar, chances are very high that those diseases are going to be very close together. So, for example, IBD and Crohn's disease or psoriasis, are, you see, they're living in closer neighborhoods than you would expect just by chance. Or all cancers, they cluster all together. So that can be used as a predictor for comorbidity. So the first thing we wanted to say is, do we have any disease that is it's comorbid of COVID? And if so, that would lead to a very fast repurposing idea, right? So because if two diseases, they are very similar, you probably can use one of the drugs to treat one another, right? Turns out that there is no disease that they have an overlapping module with COVID, but we have a lot of diseases that they are very close by. So heart diseases, brain diseases, some neurological manifestations, that is in line also with what we found previously. Okay, so that means that we really have to go for real drug repurposing, right? So for that, so first thing we have to keep in mind, right? So we have to find drugs that are going to be targeting the close, the COVID module, right? Um, and we didn't want to test only one methodology. We wanted to test different methodologies, different ideas, and see which one would be the best, or which one, or a combination of them would give us a better prediction. So we first used network proximity with 3 pipelines. We had a network diffusion with 5 pipelines, and the AI prioritization with 4 pipelines. So the proximity, basically, you have one drug here that has 2 targets, but you also have the virus that has 3 targets, right? And this is the underlying PPI, so the protein-protein interaction. And what we basically do, very short, we just count how far it is one target from the drug to one target from the virus. And we do this for all proteins that are targeted by the drug, and that leads us to a proximity score. We reshuffle the network a couple of times, 1,000 times, and that help us identify a reference distribution where we compare our result to, and that's how we can define which are the values that they are significant, right? So we want to have as proximal as we can and smaller proximity as possible. And for that, we had 3 pipelines. So the first pipeline, we used all drugs that they are available in drug bank with all their targets. The pipeline number 2, we kept only the the proteins that they are marked as polypeptide, and we disregarded all the ones that they are enzymes, carriers, transporters, or less related to the pharmacological effect, but they are more related with the pharmacological mechanism of action. And the pipeline 3, we use, instead of binding targets, we used differentially expressed genes given a particular drug. So here we have, as an example, chloroquine and etanercept. So both are drugs that they are used to treat rheumatological diseases. So they have a similar theoretical effect, but they have different targets. And interestingly, so chloroquine is very close to the COVID model, and etanercept is very far away from the model. And here we can see how their mechanism of action works, right? So in light green, we have a lot of targets that they are very close to the COVID targets, so the other green ones. While etanercept has its targets further away, where we have to put other proteins. So they act by activating other proteins, and then gets to the COVID module. We next used diffusion method. So diffusion method basically calculates, using a random walk, the probability that you have from node 1 to visit node B. So node B would be this one, and we have the node A here. So so one of them is the set of proteins that they are targeted by the virus, and the other side is the proteins targeted by each one of the drugs. And then you basically have these random walks that they're going to start from A and then go to B. And for that, we tested 3 different similarity measures, the ways that we could calculate those probabilities. So one is based on Manhattan distance, relative entropy, or Kubark library. And they work in a very similar manner as the proximity, because the closer they are, the probability that you're going to end up visiting the other node is higher. But what changes from the 5 pipelines that we have here is basically the metrics that we are using, and if we are using either the minimum or the median between those measures. And the last one is the AI-based repurposing. So we have a knowledge graph that is constructed based on drugs and their targets, virus and their proteins, and the human proteins. And we try to predict links. So that's a link prediction task, that which new diseases a new molecule can treat. So we basically use a graph convolutional network where we take this knowledge graph, we learn an embedding, so that basically for each one of the nodes of the graph, you have this embedding that they're going to be like points in the space. And we use this learned embedding and make the link predictions to identify which are given a new disease, what drug can treat that. So for that, we have 4 different pipelines, and similar to what happened in the diffusion, what changes here is the decoder. So how far we are from the COVID. So here is the COVID module. And what we want to find is drugs that they are either very close or very far away, or how local or how distant this structure can be. So that's basically the difference between each one of the 4 pipelines that we have here. Okay, so we wanted first to see if all drugs, all pipelines, they were able to produce similar set of drugs. And turns out that within each one of the pipelines, right? So within each one of the methods, they are very consistent, but they are not consistent with other methods. And that's highly expected, right? So they provide complementary results instead of giving the same result. So here what we have is it's the correlation matrix based on the Kendall-Tal, and we find that diffusion is very tied together. So they're going to predict very similar things, but it also correlates a little bit with the proximity methodology. P1 and P2, so those first one from proximity, they are very tied together, but they are very different from P3. Just keep in mind that P3 is based on differential expression and not binding targets. And all the AIs, they are also consistent with each other, but they correlate a little bit with D1. Okay, so we first wanted to see if are we predicting something that the medical community would be happy with? And by the time we didn't have any kind of experimental validation, right? So what we could see is that, and what we used was the clinicaltrials.gov, so we could see which drugs were being tested for treating COVID. So we could see that the AI method is the best one. So it has a AUC of 0.86, 87, depending on the decoder that we are using. And that's followed by P3. So that means that having the differential expression, it kind of helps more than having the binding target information. And diffusion methods, they were okay, but they were not spectacular when we compare to the others. But we wanted to see, okay, so if we have a combination of those methods, it's something gonna get better, and it does. So when we combine all of them, we have a better prediction power than when we have any of the methods by itself or any of the combination of, for example, just the proximity or diffusions are AIs, right? So the combination of all of them, it's better. So we followed for experimental validation. So where we tested 918 drugs, we found that 77 then they were effective against COVID. And out of those, we re-screened the top 200 of them. The ones with positive outcomes, and we found that 6 of them, they were effective, and 4 of them were re-screened in another human cell line. So the first screening we did is normal cells, so green monkey cells, and later they were tested in human cell line, and then we tested in another cell line and found that 6 of them, they were highly effective, 4 of them super effective, and 3, they were not in clinical trials. So how do we do this? So we have 4 categories for each one of the drugs to be fit in after the experimental validation. So the first one, we have strong drugs. So that means that you kill the virus before you kill the virus very fast and very effectively. You have weak cases where you kill the virus, you reduce the viral load, right? But it's not super effective. You have the cytotoxic ones where you end up killing the cells before you even start killing the virus. And you have the last case where nothing happens. It doesn't matter how much drug you add to the wells, they're not gonna die. So this is the list of drugs that we end up predicting, and we can see a very broad list of drugs. So we have cardiovascular drugs, we have antibiotics, we have immunomodulators, we have antiproteasomal drugs. We have many kinds of drugs, and this is what we expect, right? To have as broad as possible when not focusing on only one kind of drug. So those drugs were retested, as I previously mentioned, and those are the 6 drugs that we had that they worked quite well. So auranofin was the drug that best worked, and azelastine. However, we rank, so there are a couple of things we have to discuss now, right? So we rank drugs based on how we expect them to behave for COVID. So that doesn't mean that if a drug didn't make our final list, they would not have efficacy or they have to be excluded. No, it means that we probably don't have enough power for that. So, and that depends highly on the input data that is provided. So, for example, new hosts, virus host binding interactions, so they can be experimental or AI predicted. We need RNA-seq or microarray data coming from COVID patients. New drugs has to be added to drug bank. And mainly, the biggest issue is improved drug target identification. So I also wanted to say that this methodology is really general and allows to profile different kinds of efficacy of different kinds of drug, different kinds of diseases as well. And it could be definitely used for diseases that they are less studied or less known. So we were able to find a bunch of these drugs that could be used. There is another thing we have to keep in mind is that none of those methodologies, they enable for identifying the dose response nor the sign of the interaction. So we know that a drug is going to perturb a disease model, but we don't know if this perturbation is going to be, we're going to increase the sign of the virus, so the virus is going to be more efficacious, or the disease is going to be extinguished, right? So we don't know the direction of this interaction. And we also don't know the dose, so it might be that it can work, but it doesn't work in a bioavailable dosage that you need. So this is one example of the best drug that we have. So that's ritonavir. So this is the disease model of COVID with all the COVID proteins inside and the drug targets from ritonavir, right? So when we zoom in and see why is it working, right? So we have uh, the SARS proteins here that they are binding to the human proteins, and the human proteins are binding to some of the targets from the drug. However, we still can have some other, it can work through other mechanisms. So for example, UBC is added here so we can connect the full network. And this drug was basically ranked first by most of the AI, some of the proximity, but was ranked very low by diffusion methods. So this work was not done just by me, it's a team. And one of the things that I want to appreciate is that everybody here has a different expertise, and the combination of different backgrounds can lead to really good research. So I'd like to thank all my collaborators for being this awesome during this time. And I'm happy to take any questions. I would like to thank you all for being here and listening till the end.
So thank you, Daisy, for your nice presentation. And I'm just looking if there are some questions here. And if you can, the participants of this talk, you can write in the comments here and Daisy can see. So we have a question here from Rodrigo Ramos. And I don't know if you can see it, Daisy, but the question is, very nice presentation, congratulations. What protein-protein interaction did you use?
So we use a combination of methods. So we have 13 well-validated PPIs that we combined. And so it's not only one, but the data is available on the GitHub of the lab, and you can use the same one.
And Rodrigo also has another question, that is, what software or library did you use to plot the 3D network?
So the videos, they were done by a designer, so it's not mine. Yeah, but I think that she used Unity to plot those. But all the others, they were done in R with iGraph.
So Daisy, from your presentation, we have a lot of questions, but I would like to make some 2 or 3. Some of the drugs that you found in your research have been tested in these clinical trials. This is what I understood. Yes. And can you tell a bit more about the results and what were the outcomes from these trials, these clinical trials?
So we have now ongoing 3 clinical trials, and I cannot disclose anything about them. But some of the drugs we saw that they worked, and some we know that they did not. So one thing that is very important to mention, so chloroquine and hydroxychloroquine, they were always ranked like super high for us. And we knew that we have a very different system that goes on when we test, and like in the virtual screening, when we go to the experimental cell line validation, they work. But when you move it to the human body, we know that they don't. And that can happen, right? And this is due also to the dosage that we can put on the drug, right? We have another, so auranofin, that went really well. So the bioavailability of the drug, it has to be a little bit higher than we can take. So that's something that we always have to be really mindful. Even though we can find drugs and they can go for clinical trials, they can work, they might need a little bit more than we need, that than they, to work than we can take, and that is a big problem.
Mm. Yeah.
So and I believe that for these methodologies to be really broadly used for any kind of disease, we still need a second step that
Mhm.
is predicting the dosage for those drugs.
Yeah.
So I highly
And
advocate for that.
sorry.
I highly advocate for someone helping us identifying the
Ah,
dosage for the drug.
okay. And another questions are more, it's not a technical question. This is a very interesting treat diseases, but it really needs very powerful computational resources. And I team with many distinct education, right?
Yeah.
So, if you can tell us a bit about your team that you showed in the last slide, and if you can say something about the computational resources, only to see, because this really needs personal staff and an infrastructure very powerful, right?
Yes. So for the proximity and diffusion methods, you don't actually need a lot of computational power. So depending how you implement the code as well, right?
Mm.
But proximity doesn't really require a lot as much as the AI. I don't want to say that they're gonna run in a personal computer.
Mm.
All of them they needed cloud. All of them they needed like to run in the server. So, but the AI part, it really needed a bunch of computers and GPUs and like a whole structure. And the person that did all of that was Marinka Zitnik. So she's a professor here at Harvard, and she works with AI and drug repurposing methodologies and polypharmacy. So she had a lot of experience, and I think that everybody in the team, they had a really, like, everybody was really important because we were meeting every day for like 3-4 hours every day for a couple of months, and discussing a lot of results and what everybody has an input and things like this. And that was only, like, we could only achieve this because we had a very multidisciplinary team. And I think that's one of the best things that happened during the pandemics. If something was good during the pandemics, was that people were able to acknowledge that we need other people working with us, and teamwork is always better than working alone. Um, Independent of people might say like, oh, it's boring to work with other people, but we need to work with other people because we learn so much.
Mm.
So for example, all the AI part, we wouldn't be able to do by ourselves if we didn't have Marinka, that she's an expert on that. Uh,
Mm.
a similar part that I didn't show like in depth. So we had a clinician on the team that would also guide us, telling like, oh, this drug doesn't work. This drug is like,
Mhm.
it's topical. So another drug that we had that worked quite well was camphor. That is the same one that works, like it's Vicky that we take when we have a flu or something like this. So that ranked really high for us. But how are you gonna think that Vic's gonna work? So you need to think about the how the applications are being made, what's the viability. We also had the experimental team that was amazing. Um, they were really good, and they gave us so much insight on how things should work. So for example, and that's another thing that is important, right? So the whole the whole screening that they did. So we used 900 drugs, but the whole screening we had 8,000 drugs. That is a second paper that we are working on. So from this 8,000 drugs, they manually identify which are strong, which are weak, which are cytotoxic. So part of what our team also did, the part that I did, was basically create a classifier for them that you just give a drug and I can build a tool that you're gonna have like already like classified 8,000 drugs. Doing by hand, it's a massive work. So that is also important why teamwork is important, right?
Yeah.
Yeah.
Yeah, we have two more questions here from Marcelo Reis from Butantan. Thank you for your nice talk. What could be done to narrow the gap between PPI-based prediction for a single cell and the efficacy in the organism level?
Okay, so for the what we often do to filter and narrow down the tissue specific PPI or cell specific PPI is understanding which are the proteins that they can be expressed there, and we filter based on that. However, if you really want to have a specific PPI, and you would have like as much money and time as possible, right? Um, all the experiments should be done already focusing on that particular cell. But that is almost that would be very hard to do because you cannot generalize, right? That's going to be really specific to one cell type. Um, and it's a similar way when you think about the organism level. So we have the whole PPI that we use as the full organism, but we know that it should be taken into account how they relate. So each protein relates to each other in each one of the cell types or the tissues, and then you generalize this for like a multi-level step. Um, but that would require so much computational power that we are not doing this yet.
And the other question from Marcelo is about multi-scale models, which describe the interplay between tissue-specific PPIs. This could be a possibility.
So there are some papers that they really show how like the PPIs change according to the tissue and how they interplay using multi-layer networks, right? So that's how people have been modeling that. But I don't see as much work on that as I would like to see. But that definitely would also help improving the previous part, right? So understanding the efficacy of a drug given the whole tissue specificity network.
And Daisy, I'm very curious to know if you know some groups or some people that are working in this direction in Brazil.
So on the top of my head, I can only remember Elder Nakaya that he was working also with similar things, but I think it's still very different from what we are doing here. So that is definitely a field that we would need more people working, and mainly in Brazil, right? So the same methodologies they can be applied to many tropical diseases, right? So, for example, yellow fever, dengue, or Zika, like they, apart from dengue, all the others they don't have any treatment, right? So you have the vaccine, but you don't have a treatment, and that could help us identify possible treatments that are already out there.
Yeah, yeah, sure. I don't see any other questions here, and so I would like to thank you again, Daisy, for having accepted our invitation. We hope to see you in Brazil And I hope that we have here better conditions to receive young scientists like you. And so thank you, and we finish this talk.
Thank you so much, Mia, for having me here. It was a pleasure. And yes, I also hope to at some point go back to Brazil. I think that the science in Brazil has a lot of potential. So definitely it's a place that I would consider. And I just want to mention that we will be hiring for postdocs soon. So if someone wants to apply for the lab, just shoot me an email. We're gonna have something on the web page probably soon.
Oh, there are good news.
Yeah.
Thank you so much.
Thank you. Bye.
Bye.